Hello, my name is Pál Weihe. I'm a medical doctor in the Faroes healthcare system. For the last almost 40 years, I have made research on how marine pollutant can have impact on the human health. And I'm going to tell you the story today in short. Thank you. On this first slide, you see that analysis of museum samples reflect the increase in mercury emission and releases since the mid-19th century associated with anthropogenic activities, that mean man-made activities. On the next slide, you see that concentrations of mercury in the Arctic marine animals today are about 10 to 12 times higher than in pre-industrial times, that before 1800. This means that on average about 90% of the mercury in marine predators such as seabirds, seals and whales is anthropogenic in origin. The timing of the increase suggests that 19th and 20th century emissions from Europe, North America and Russia were responsible. The pilot whale has served the Faroes well for many hundreds of years and has likely kept many Faroes alive through centuries. But the times and the environment are changing. Now the pilot whale is heavily polluted with persistent organic pollutants and methylmercury. The mercury levels in whale meat have not declined over the years. However, the most of the persistent organic pollutants have decreased due to the regulation in the Stockholm Convention. Some fluorinated substances have diminished due to stop of production, and others are increasing. Prospective cohort epidemiological studies undertaken in the Faroe Islands have demonstrated that children exposed to methylmercury before birth exhibit decreased motor function, shortened attention span and reduced verbal abilities, diminished memory and reduction in other mental functions. Follow-up of these children at the age of 4 up to 20 years indicate that these effects are permanent. In the Faroes, we have followed approximately 3,000 children from birth in 6 different cohorts. The first was established in 1986 and the last in 22. On this slide, you see the timeline for the cohorts. The total time of examination and re-examination are approximately 40,000 hours, and we intend to continue the research work in the years to come. In our prospective studies over this 40 years, we use different methods, and all of them are indicating negative effects on the brain of the children. In neuropsychological test domains like attention, language, and memory were negatively affected by increasing mercury in the umbilical cord. In our cohort, we saw a significant difference in reaction time at age 7 years. The highest exposed had longer reaction time than the less exposed. Neurological optimality score showed a negative association too. The higher cord blood mercury, the lower score in neurological optimality. When measuring electrical signals in the brain, so-called evoked potentials, we found prolonged 3-5 intervals on brainstem auditory evoked potentials associated to higher mercury in children at age 7 and 40 years of age. This indicates that mercury is inhibiting the electrical signals in the brain. When the children at the age of 15 were examined in a MR scanner, tapping with the left hand, we found an increased activity in the brain of the highly exposed, indicating difficulties in processing a simple stimulus. After 3 decades of research in this child cohort, the conclusion of neurotoxicity related to mercury in the brain of the Faroes are the following. Neurobehavioral effects, most clearly on attention, memory and language, but also visuospatial and motor functions. Parallel neurophysiological findings. Prenatal, generally, exposure was more toxic than postnatal. Results from 14 and 20 years of age indicate permanent effects. An expert panel convened by the US EPA in 2011 concluded that methylmercury is directly linked to acute myocardial infarction and to increased cardiovascular risk factors such as atherosclerosis, decreased heart rate, variability, and to a certain degree hypertension, as you see on this slide. You can see an association between mercury and blood pressure. The beneficial effects of seafood nutrients such as omega-3 fatty acids, including DHA, may mask some of the adverse effects of methylmercury. We found no evidence that selenium was a significant protective factor against methylmercury neurotoxicity. Selenium intake can therefore not explain the significant benefits associated with fish intake, as we have seen in our studies. Preventive methods are needed to address methylmercury exposure rather than selenium intake, while maintaining a high fish seafood intake. The concentration of mercury in whale meat has not changed, but the concentration in mother's blood has declined significantly. The plain explanation is that Faroes pregnant women do not eat whale meat anymore. The pups have a long half-life in humans, for some substances 10 years. Therefore, the reduction regarding the sum of PCB is remarkable, indicating that contaminated blubber is no longer a part of the mother's diet in the Faroes. A new group of substances is the polyfluorinated compounds. We have analyzed the serum concentration of this fluorinated compound in the Faroese whale meat consumers. The concentration found are comparable to those reported elsewhere, for example in the US. However, PFOS and PFNA concentration correlated well with the frequency of pilot whale dinners and with concentration of mercury and polychlorinated biphenyls, also PCBs. We suspected that these substances could have a negative impact on the immune system. In our cohort studies, we have seen that PFC were highly associated with reduced humoral immune response to routine childhood immunization, age 5 and 7. Since around 1980, pregnant women have been advised to limit their consumption of pilot whale meat and blubber and to avoid mercury exposure. In the 89 recommendation, adults should not eat pilot whale more than 150-200 grams of pilot whale meat and 100-200 grams of blubber per month. The toxic effects have been documented at progressively lower levels of exposure as increasingly sensitive and sophisticated diagnostic instruments have been applied over the past 4 decades. The safe threshold has declined. The current reference dose used by US EPA is based on effect data from the Faroes in 1997. However, recent findings indicate that even lower effects with neuropsychological methods, neurophysiological methods, and in groups with genetic predisposition. In 2008, the first medical officer of the Faroes, in agreement with the Danish Board of Health, went a step further and recommended that pilot whale was no longer used for human consumption in the Faroes. The Faroese society is affluent, and the availability to seafood low in contaminant is easy and relatively inexpensive. We have not found any additional healthy substance in pilot whale meat and blubber which not can be found in seafood caught, farmed by Faroes in in large quantities, for example, the cod, haddock, mackerel, herring, and salmon. My conclusion is simple. After almost 40 years of research on health effects among pilot whale consumers, we have not identified any beneficial effect which cannot be obtained consuming the fish species we catch and export. On the next slide, you see the team from different countries, from Japan, from US, from the Netherlands, in our research station. Thank you, and perhaps I can answer a question via the telephone, or you can send it to me as a mail. Thank you.